Hello and welcome back to Crash Course. Today we're going to be continuing to look at the neurosensory system. In part 1 we did take an in-depth look at the anatomy of the nervous system, and in future videos we are going to continue to look at other aspects of the nervous system, including the central and peripheral nervous system and the different tracts. But today what we're really going to focus on is the embryology of the nervous system alongside the cranial nerves and the visual and auditory systems. So let's start with the embryology of the nervous system. The nervous system is derived from the ectoderm alongside the skin, and at the beginning of week 3, a visible line or streak of cells forms on the epiblast layer, and this streak essentially deepens to form a groove or pit at the end of it at the cranial end of the primitive streak. So, as we can see on this diagram, we have the endoderm, the mesoderm, and the ectoderm, and the ectoderm is where this primitive streak is almost rising from in terms of this diagram, and it's ascending all the way to this cranial or rostral end, where it will form the primitive pit or node. The ectoderm cells tunnel from the primitive node longitudinally and cranially, and this forms a hollow tube known as the notochord. So this is what we're looking at normally, and then eventually the notochord is what's going to be tunneling here longitudinally and cranially, and the notochord becomes the nucleus pulposus of the intravertebral discs. So in those IV discs you've got that nucleus pulposus in the centre, and that's what your notochord is going to become. The notochord, however, does induce other changes within the embryo, and growth factors from the axial mesoderm act upon the ectoderm. So factors released from the mesoderm act on the ectoderm, and this causes the ectoderm cells to differentiate into neuroectoblast cells, and this is essentially the neural plate. So as you can see, the neural plate is forming around this primitive groove and primitive pit, and this is due to the release of growth factors from the axial mesoderm on the ectoderm. So the cranial end of the neural plate will become the brain, and the caudal end will become the spinal cord. As the neural plate forms, it sinks in the midline to form the neural groove, and there are folds either side of the neural groove that by day 25 meet in the middle. And essentially, this is the transformation of the neural groove to the neural tube, and the neural tube cells will make up all of the matter of the spinal cord, as you can see here. So this is a neural groove, eventually folding in, and the neural plate forms, and this is the neural tube here by day 25. Neural folds merge in the middle of the embryonic disc first and spread cranially and caudally, and an error here in terms of pathology will cause spina bifida. This whole process overall is known as neurulation, and like we say here, we've got that folding merging in the middle of the embryonic disc first and then spread cranially and caudally. So in the neural tube we have a dorsal portion, and this is also called our alar plate, and this is going to form the sensory area of the spinal cord. So associate the dorsal or alar plate with the sensory area, and the ventral portion or the basal plate will be the motor area. So what's really important is our dorsal ganglia in our spinal cord carries sensory information, and our ventral portion of the spinal cord carries motor information, and therefore this is a clear link to embryological deprivation. So the alar plate, as you can see, is on this side, and the basal plate is down here, appropriately being dorsal and ventral. So during neurulation, some cells break off from the dorsal aspect of the neural tube, and we call these neural crest cells. So from the dorsal aspect, from the back, we get these neural crest cells, and these become the sensory neurons of the dorsal root ganglia. The neural crest cells extend axons both peripherally and centrally, which is why we find pseudo-unipolar neurons in the dorsal ganglia. We're one of the only mammals to have pseudo-unipolar neurons. And on the other hand, motor axons just extend from the neurons in the alar plate. So quite simple. So from the dorsal aspect, we get these neural crest cells, these are the sensory neurons, and from the alar plate, we get these motor axons, which just kind of derive or extend from the alar plate itself. So moving on to the spinal cord, as the mesoderm develops, it can become segmented, and axons extend peripherally from the alar plate, so motor as we said, and neural crest cells sensory, and grow into the adjacent mesoderm. And when this happens, this forms what's known as a somite, and each pair of somites will turn into a myotome or a dermatome, and you'll be familiar with the dermatome map clinically and its clinical relevance. So the segmentation of the neural tube explains the segmentation of the spinal cord and why different muscle groups are associated with different spinal levels, which is why when we feel sensation, it refers to different spinal levels. So this, for example, here is a somite. So next, thinking about the development of the brain, at the cranial end of the neural tube it becomes pinched to form these three distinct regions, and these are the forebrain or the prosencephalon. Then we have the midbrain, which is a mesencephalon, and then the hindbrain, the rhombencephalon, at the bottom. And the walls of the vesicles will become the brain tissues, and the lumen will become the ventricles and the spinal canal. So during week 5, the forebrain and hindbrain both subdivide. The midbrain doesn't, so the midbrain remains, but the forebrain, the prosencephalon, and the rhombencephalon both subdivide. So the forebrain divides into a telencephalon and a diencephalon here. And then we've got the midbrain, which just stays the same. And then the hindbrain further subdivides into a metencephalon and a myencephalon. Essentially, these all go on to become different regions of the brain, whether this be the cerebral cortexes, the basal ganglia, the pons, and the cerebellum, or the medulla oblongata. But essentially, all of these different vesicles or regions will go on to become different parts of the brain. So, it's all about the lumens. So, the lumens of the telencephalon become the lateral ventricles, the lumens of the diencephalons become the third ventricle, the lumen of the midbrains becomes the cerebral aqueduct, and the lumen of the metencephalon becomes the upper fourth ventricle. And then the lumen of the myencephalon becomes the lower fourth ventricle. So, what's really important about these form the ventricular system of the brain. So, further development. As the development continues, the cerebral cortex becomes folded into sulci and gyri, and by 26 weeks the lateral sulcus is evident, and by 30 weeks the cerebellum becomes distinct. The embryology of the nervous system is quite confusing, so take it slow, take it slide by slide, and do try to understand it. Divide it into the brain and spinal cord to allow you to understand it bit by bit. So next we have the cranial nerves. So of course, hopefully we should know these already. The 12 pairs of cranial nerves, so olfactory, optic, oculomotor, trochlear, trigeminal, abducent facial, vestibulocochlear, glossopharyngeal, vagus, accessory, hypoglossal. So do try to get them rolling off your tongue one by one. And 10 out of 12 of these cranial nerves do arise from the brainstem. And the only ones that don't are the optic and olfactory nerves, which arise from the cerebrum themselves. So the cranial nerves relay information between the brain and parts of the body. So primarily to and from regions of the head and neck mainly. So as I said, we've got OOO, TT, AF, VGV, AH. So, and then we've got some say marry money, but my brother says big brains matter most. And that's just a little anagram to allow you to remember the way in which each of these cranial nerves works, whether they're sensory, motor, or both. So matching those up, we can think of ways in which we can remember them. So the top one is ooh, to touch and feel very good velvet are heaven, or I'm sure you have your own ways of remembering that you can find online. Alternatively, you have the bottom ones, which can be some say marry money, but my brother says big brains matter most, as we've just said. And this is a really, really easy way to remember what each of the nerves function is, whether it's sensory, motor, or both. Another key aspect is understanding where the cranial nerves originate from and where they exit the brainstem. So, for example, the olfactory nerve, cranial nerve number 1, is responsible for sense of smell. It originates from the cerebrum and exits via the cribriform plate. I'll allow you to work through these on your own. You will learn your own ways of remembering these, but they're really important as it's a really easy exam point scoring question. But please do be aware that several of them originate from the same place, several of them exit at the same place, and actually some of the nerves are self-explanatory in terms of what they do. So simply learning the eye nerves, so optic, oculomotor, and abducent already rules out 3 of them in terms of what their function is. So moving on to vision in that case, simple eye movements, elevation, depression, adduction, assuming you're moving towards the nose, abduction, assuming you're moving out towards the ear, intorsion, and extorsion. So to put that into perspective, there are 6 extrinsic voluntary eye muscles, and they run from a tendinous ring at the posterior wall of the eyeball, and this is called the annulus of Zim. So, simple thing to remember about the eye is the SO4 LR6 EE3, and if you remember this, you'll remember everything you need to know about the eye, really. So, superior oblique is innervated by cranial nerve number 4, lateral rectus is innervated by cranial nerve number 6, so the abducent, and everything else is innervated by cranial nerve number 3, so oculomotor. To label those, you've got the superior oblique, the superior rectus, the medial rectus, lateral rectus, inferior rectus, and inferior oblique. But what's really important is understanding what each muscle does. So, for example, the medial rectus is involved in adduction, so moving towards the nose. Your lateral rectus is therefore doing the opposite. It's abduction, it's moving towards the ear. Then we have the superior rectus, which is involved in elevation, adduction, and intorsion. We then have the inferior rectus, which does depression, adduction, and extorsion. Then we've got superior oblique, which is depression, abduction, and intorsion. And inferior oblique, elevation, abduction, and extorsion. Unfortunately, it is a case of just learning what each of these muscles do. But be logical when you're thinking about simple eye movements. So, for example, looking up, you'll combine the superior rectus and inferior oblique. Elevation is what you want to achieve. Abduction and adduction cancel each other out, and extorsion and intorsion cancel each other out as well. Use the same principle when thinking about looking down. Labelling the eye, so it's 3 layers: fibrous coat, vascular pigmented coat, and a nervous coat. And these overall are the 3 coats surrounding the eye. So the fibrous coat is made up of the sclera and the cornea. The vascular pigmented coat is made up of the choroid, plexus, and the ciliary body. And the nervous coat is made up of the retina and the blind spot and the optic nerve as well. There's also this macula lutea here, which is just a little bit of a central depression called the fovea centralis. So the pathway to the visual cortex, again, this is something you really do need to know about. So the optic nerve runs to the optic chiasm. The optic tract then runs from the optic chiasm to the lateral genicular nucleus to the thalamus. And then from the lateral genicular nucleus, fibers pass posteriorly to the visual cortex within the occipital lobe. So, putting that into a diagram, it all looks like this. For first year, in terms of anatomy and physiology, you don't need to know about these. These are pathological consequences if the cord is obstructed, but for second year you will need to know about these. Understanding this diagram is absolutely crucial in terms of where the light comes from and how it passes back to the optic chiasm, to the lateral genicular nucleus, and then lastly to the visual cortex in the occipital lobe. There are different types of photoreceptor cells, and these are specialized neurons which allow phototransduction, and they absorb photons, triggering membrane potentials, converting light into biological messages. And you can either have rods or cones. So rods are responsible for vision at low level light in comparison to cones, which are active at higher levels of light. Rods have no colour vision, but cones do. And rods have a low spatial acuity, whereas cones have a high spatial acuity. Now to finish, we'll look at the auditory system, and essentially to have a quick label of this is a helix and the auricle. This is the lobule, so that's all to do with the external layer. Then as you move in, you've got the external acoustic meatus of the ear canal. You've got the tympanic membrane. Then we've got these 3 bones here. These are the ossicles, so the malleus, the incus and the stapes. Then we've got the semicircular canals, the cochlea, the vestibular nerve, and the cochlear nerve, which together become cranial nerve number 8, the vestibular cochlear nerve. Being able to label this is really important, but also be aware of the functional divisions of the ear. So you've got the external ear, the middle ear, and the internal ear. So the auditory system is comprised, as we've said, of the outer, middle, and inner ear, and the main nerve here that we're really bothered about is cranial nerve number 8. So the process of hearing, of course, begins with some kind of sound. Sound is initiated when an event moves and causes a motion of vibration within the air, which our ears detects as a stimulus. And that sound wave is then transmitted through 4 separate mediums, through air, mechanical, liquid, and neural functions, all the way to the brain. So, in terms of functional divisions, we've got the external ear first, or the outer ear, and this includes a portion of ear that we can see, in other words, the pinna of the auricle of the ear canal as well. So the pinna is that outer cartilaginous structure which detects the sound waves travelling in. The ear canal is the inner 2/3 in the temporal bone, and then the outer third is cartilage. So although the shape of the ear, each ear canal varies, in general the canal forms that elongated S-shaped curve, and it directs those airborne sound waves towards the tympanic membrane, or our eardrum. Then we have the middle ear. So the middle ear is composed of the tympanic membrane and cavity, the ossicles, the eustachian tube, and the mastoid, so quite a large portion of the ear. The tympanic membrane essentially is that eardrum, the grey-pink, thin layers of living tissue, which is very sensitive to sound waves. You've then got the eustachian tube, which acts as an air pressure equalizer and ventilates the middle ear. And then, of course, we have the ossicles, the stapes, the malleus, and the incus. And then attached to these ossicles are 2 tiny muscles, the stapedius and the tensor tympani muscles. The mastoid is a bony ridge behind the auricle that connects the middle ear, and it can get infected, causing mastoiditis. Lastly, we have the inner ear, and this is composed of the cochlea and the vestibular system. And the vestibular system is really responsible for our balance, and it's made up of 3 semicircular canals located within this inner ear, and it's all about the head position and gravity and maintaining balance, and it's all linked into the vestibular cochlear nerve. In the inner ear, you've also got the cochlea, therefore. This is the hearing part of our inner ear. It's spiral shaped and similar to the shape of a snail, that should say. So the cochlea is composed of 3 fluid-filled chambers composing of perilymph and endolymph, and essentially these allow the passage of the sound waves through to the neural component here, which is cranial nerve number 8, transferring this information to the brain. So to follow this through, we hear the sound in the outer ear, it passes through to the middle, then to the inner ear, all the way to the brain via the 8th cranial nerve. That's everything for this video. Sorry to rush a little bit towards the end. I do just try to keep these videos under 15 minutes the best I possibly can. If you do have any questions, please feel free to contact me, and I hope you did find this video useful. Thank you for listening.